Hello, everyone, and welcome to our ADMET Predictor webinar on supporting medicinal chemistry programs. I'm Michael Lawless, Senior Principal Scientist at Simulations Plus. In today's webinar, Dr. Eric Jamois, Director of Business Development, and Dr. David Miller, Director of Cheminformatics, will be presenting tools in ADMET Predictor for analyzing compound collections, selecting subsets of libraries that are enhanced for selected properties, creating a QSAR model to predict IC50 values, and generating analogs of lead molecules. An opportunity to ask questions will follow the presentation. Please enter your questions by using the questions pane on your GoToWebinar control panel. This webinar is being recorded for playback at our website, www.simulations-plus.com. I'll first give a brief overview of ADMET Predictor's modules and discuss a few of the tools in the MedCam Studio module. Eric will then present an analysis of commercial compound collections. Afterwards, David will talk about QSAR model building and de novo design. But before I start, I'd like you to respond to a following poll. So let me launch that poll. And I will give you about 30 seconds to answer that. So if you could start clicking on that, and I see we are getting some responses, and we would certainly appreciate that. Okay, about 10 more seconds. Looks like they're converging. Okay. All right, well, thank you very much. That's going to be good information. So if I go to my second slide, this is the 6 modules in ADMET Predictor. So we have the physical, chemical, and biopharmaceutical, metabolism, toxicity, HTPK simulation, ADMET Modeler, and the MedCam Studio module. The physical, chemical, and biopharmaceutical contains models to predict things like log P, log D, pKa, permeability, solubility, etc. The metabolism model contains models for predicting CYP metabolism, UGT, AOX, and esterase metabolites. Our recombinant CYP models are kind of a tiered approach. So we predict whether a compound is a substrate or non-substrate for a certain isoform of cytochrome P450. If we do predict it as a substrate, then we predict the sites of metabolism on the molecule, and then we also display the metabolites in our MedCam Designer program. We also calculate things like Michaelis-Menten kinetics, Km, Vmax, and clearance for these recombinant CYPs, and we can display those also in MedCam Designer. The toxicity module contains toxicity modules such as hERG liabilities or hERG binding and immunogenicity. The HTPK simulation module stands for high throughput pharmacokinetic simulations, and by default it will use predictions from the physical chemical module and the metabolism module. With the metabolism module, it will use the human liver microsomal clearance or rat liver microsomal clearance. The module also has the ability to take your own experimental data. So as you synthesize the compounds and you collect things like solubility and maybe hepatocyte clearance, you can input that and use those experimental values in the HTPK simulation module. And it will predict things like fraction absorbed, oral bioavailability. We also have a module that will predict the dose required to reach a total plasma concentration. You can also display CP time curves, calculate Cmax, and a number of other PK parameters. The ADMET Modeler package is actually what we use to create these artificial neural network ensemble models in these 3 modules. And David's going to show an example of building a model with ADMET Modeler. The focus of this webinar is the MedCam Studio module. And on this next slide, I just show a few of the tools involved in the MedCam Studio module. So one of the most powerful ones is the ability to organize the compounds into scaffold classes. So this will take a set of compounds and cluster them into classes that contain a common scaffold. Once you define the scaffold, it's very easy to decompose the R groups and then drill down into the SAR information there. The module also has the ability to create your own libraries or data sets or databases, and you can perform substructure and similarity searching on those data sets. It also calculates structure property correlations using keys. You can use max keys or these extended keys also. The match molecular pair analysis is really an excellent tool for looking at trends in the data and finding compounds that are very similar and then analyzing an endpoint such as solubility. It also allows you to analyze activity cliffs, and we use that quite a bit when we're building models to look at compounds where maybe there's a small change in structure of the molecule, but it has a very large change in the property. You can also characterize compounds using structure and property alerts and design novel compounds, and David will talk a lot more about that. So before we start with Eric's presentation, I just want to run our second poll, which we appreciate if you could answer. This is, are you currently using computational tools to support your medicinal chemistry programs? And I'll launch that. If you can say yes or no to that, we would appreciate it. So I'll give you about 20 more seconds on that poll. All right, about 3 more seconds. All right, thank you very much. And it's my pleasure to introduce Dr. Eric Jamois. And I'm gonna go ahead and make you the presenter. So if you can take over, Eric.
Yeah, I will share my screen, and hopefully you can see my screen.
Yes, we can.
ADMET Predictor. Yep, sounds great. So good morning, good afternoon, good evening to everyone, depending where you are. I'm going to take you through a couple scenarios of using ADMET Predictor on a data set from a company that I was previously associated with called Lawless. That's a company that's involved in the design and synthesis of topological mimetics of natural products. These are synthetic compounds that are actually inspired from natural product scaffolds. So they were kind enough actually to provide me with this data set to be used as examples of the kinds of things that that they do. So I'm going to read in a file of 500 compounds, which are pyrido-oxazepinones and variations on that theme, which you can see here. So something that is not really obvious to the eye right now, that there's actually 4 subclasses within these 500 compounds. And what I propose we do first is that we use some of the clustering tools that are available within ADMET Predictor to show you these classes. So under chemistry, we can generate classes, and there's a number of options that you can use to generate classes. Here we're dealing with essentially core and R group type systems, so we will use the ring anchored systems option to generate classes. And what is happening here is that there's a new column that is being created that you see as class list, which has basically assigned a class membership to every compound. So compound 1 is in class 2, and also in parentheses it's giving you the number of compounds that are within each class. This is actually a lot easier to see. Right now I'm in the compounds tab on the bottom left. If you go to the classes tab, it's actually a lot easier to see the makeup of that set. There are 4 classes from 1 to 4, with the class sizes indicating that the first class has 299 compounds, the second class is 132, 59 in class 3, and 10 in class 4. So what you see is actually the scaffold is in blue, and then you have a so-called representative structure that is depicted here, which actually happens to be the lowest molecular weight compound within each class. So you can see the first compound is a pyridoxazepinone. The first class is pyridoxazepinone, and so is class 4 with a variation on the regiochemistry on the pyridine ring. And class 2 and 3, as you can also see, are essentially the sulfonamide analogs, with also some regiochemical differences on the pyridine ring. So right now, these compounds, I mean, they were drawn or oriented by whichever way the chemists drew them. What I propose is that we orient them in a more consistent fashion, so that all of this becomes a little bit easier to see. In order to do this, we can use the MedCam Designer, which is our free viewer application. And actually, it does a lot more than viewing, because you can calculate ADMET properties within that software. And what I propose we do is that we select our first scaffold here from class 1, and we can do a quick cleanup of the geometry, so it looks better. And being an optimistic person, I would probably like to see the carbonyl pointing up. So if I flip it vertically, then it appears in an orientation that I think most people would think is pretty good. Now, if we select all of the compounds, we can align all of them based on common scaffold, and that will bring the compounds in a common orientation that I think we can agree looks better than what we had before. And then when we're happy with that, we can transfer them back to the classes tab. So now we have these compounds, and now it becomes a lot easier to see that regioisomeric differences that I mentioned between classes 1 and 4 and classes 2 and 3 becomes a lot easier to see. The compounds themselves actually are not yet aligned. Again, they were drawn, they're oriented by whichever way the chemist actually drew them at the time they were entered. That's something we can actually very easily do by right-clicking here. We can align all the compounds in the Compounds tab on these scaffolds. So I just propagated that, and now all our compounds are actually oriented in a systematic way. The other thing that we can do is to highlight those cores so that the common cores are highlighted in blue, and then we have our groups that are in black. So I think we often underestimate how visual what we do is, and how important it is to have a really good visual handle on what we're doing. So one of the things that ADMET Predictor is really good at is actually looking at properties and profiling compounds according to ADMET properties. So that is what I suggest we do next. And so under data here, we can calculate ADMET properties for all of these compounds. So I'm just going to go ahead and calculate ADMET properties for these 500 compounds. Hit the calculate tab, and we have about 500 compounds. So I mean, literally, it takes only a few seconds to calculate ADMET properties for these 500 compounds. So what I want to show you is that there's actually different kinds of data within the software. There's just standard numeric data, which is literally black on white over here. There is also extended numeric data, which is, for example, for solubility, you do have the ability to look at solubility as a function of pH, for example. So there's many properties that are like this, where you can actually again have access to a lot more information about the compounds. There is text information as well, such as ADMET code, and then there are also what we call star plots, which are essentially a visual profile for a given compound on a set of properties. So for example, on the toxicity profile here, it's giving me a sense on hERG and other toxicity related parameters, what the profile of that compound actually is. And the length of these wedges actually are representative of the value for this particular compound relative to the range for that property across the entire set. Okay, so and we can look at this for everything. So for SIP substrate, for example, you can see that it's giving you essentially the how promiscuous a particular compound may be against the SIPs, how many of the isozymes that it's likely to be picked up by. So that is, of course, interesting at the level of every compound. It's actually a lot more interesting in many instances to look at it at the level of classes. So, and basically what we have here is we're now looking at information at the level of each chemotype. So for the properties, for the numeric properties that I mentioned earlier, we're actually looking at class averages. So for water solubility, which is the S plus SW column here, it would tell us that class 1 has the largest solubility. Those are the most soluble compound, and class 4 are the least soluble. And it's actually really interesting to look at things at the chemotype level, because then we can make informed decisions about, say, how many compounds in a particular class you want to synthesize based on, assets and liabilities of these particular classes. So again, and what we're looking at here is this entire set of compounds has been exemplified. So all of these compounds have actually been made, right? So what may be interesting to do is to retrace the steps that were involved in deriving these subsets from potentially much larger data sets. So for example, for class 1 here, we may be interested in knowing, well, again, what steps were involved in deriving this subset of 299 compounds from a potentially much larger virtual set. So what we can do here is go to the select this class 1 and go to the R tables, and we have the R group decomposition for these 299 compounds. So we see that the R1 spans 2 choices, R3 spans 13 choices, R3 4, and R4 25. So if we do a quick combinatorics here, you will realize that 25 times 4 is 100, times 13, that's 1300, times 2 is 2600. So we potentially have 2,600 compounds that can be made based on this score and R group motif. It's a slight simplification because, as we know, there can be some incompatibilities between the introduction of certain R groups, but as a first approximation, we can consider that there's potentially 2,600 compounds as a virtual set here out of what the company actually exemplified. And what we can do here is use this option called explode products, which will actually enumerate the entire 2,600 compounds in an SD file. So going to specify a file name here, a class 1, we're going to call it all 2,600. and we can quickly enumerate those 2,600 compounds. And what I propose we do now is we're going to bring in these 2,600 compounds. So we're going to open this file, which is at the top there, and bring it in. So what we see that is pretty interesting is that the compounds that were actually exemplified, that were selected to be made, have an EDX identifier in the product in class. The ones that are not, that do not have anything that for which it's blank, have remained virtual. So those compounds were never actually made. So what I propose we do is that, again, we calculate ADMET properties for these compounds, and so we're going to do that. So now it's going to take a little bit longer because, well, we have 2,600 compounds. Before we had 500, so it takes a little bit more time. It's still pretty quick. Right now it's going on my simple Dell laptop. It's going at about 150 compounds per second. We're also working on a parallel version of the descriptor calculation. So if you have a multi-core, multi-threaded machine, you will soon be able to benefit from that hardware boost. So again, it's even on again a relatively simple machine here, it only takes actually less than a minute to calculate these properties. This is something you can also do in batch mode, in command line mode, or using some sort of pipelining tool in order to perform these kinds of calculations. So, so we have the same properties that we had previously. What I'd like to show you is if we select a property like solubility, for example. So let's go ahead and show you the distribution of predicted solubility for these compounds, and I have to go down here to select the solubility parameter. This typically looks better on a log scale, so I'm going to activate the log function here, and this is the distribution of log of S plus SW, which again is our solubility parameter, across the entire set of compounds. Now, if we select the compounds that were exemplified, so we're going to do this using Control F, gives you the search option. We can search for EDX, which is the identifier, across all compounds. And now the exemplification set, what was actually made, is selected. And using this option here, update chart, we can see a representation of the exemplified set compared to the total set of compounds. And what we can see here is that in terms of solubility, they are better than the distribution would suggest, and that's of course because there was some human intelligence here that selected these compounds, preferentially as the exemplification set. We can take another property, let's say ADMET risk, for example, which is over here. We don't need a log scale for that, and show that again, when we take our ADMET risk metric, that the compounds that were exemplified again are the ones that overall have lower risk compared to what's available in the entire set. So, so the next question is, well, that's very nice, but how would you actually perform this kind of selection within the software? So here we're really only dealing with one class of compounds, so I'm going to retrace sort of similar steps to what we did before, use the generate classes option, except that now we just only have one big superclass. So it's very simple. If I go to the classes tab, we only have one scaffold, which we define as a scaffold using the MCS function. And then reflect that back in our compounds tab, so we can see our core and R group. Again, this is pretty similar to what we have done before. So what we're going to do here now is actually I'm going to show you how you can design your own subset of compounds using whatever set of properties that you would like, how you would design a biased subset according to, again, whatever parameter you would like. I'm going to use ADMET risk here to do this. So I'm going to the RTables function, which again gives me the breakdown of the compounds in terms of core and R group, and using the functionality within RTables, I'm going to select a subset. So we're going to use the find best subset option. And there's a number of parameters we can adjust here. We're going to have 3 unconstrained R groups, and we will select 300 compounds. That's close enough to the 299 that were selected. And this is the function against which you want to optimize the subset. So we said we wanted to use ADMET risk, and I will show you later what that's made of. In this particular instance, as we saw, smaller is better. So we leave that larger attribute option unchecked and say OK. And now we can see that we have achieved a selection of actually 325 compounds because it's a combinatorial set. So we have a combinatorial constraint. And it's showing us in this R group function here, it's showing us that for R1 and R3, the substituent is hydrogen, and for R2 and R4, it's actually spanning the entire 13 for R2 and 25 for R4. And 25 times 13 is 325. Okay. The one thing we can do also is show this selection in the R group analyzer, which I'm going to show you right now. Let's make this large and select our ADMET risk property, which is the one we optimized against, okay, for R2 and R4. And as we can see, these are all pretty nice. Everything is green. We're on the good side of the spectrum for ADMET risk. So one question is, what question could be, how good is that subset against a parameter that we have not optimized or we don't think we've optimized against? So let's say I want to take a look at the distribution of solubility like I did before. And if we select S plus SW to see that on a log scale, and what we see is that, well, this set is also biased with respect to higher solubility compounds. And I think most people would say, well, how could this be? You didn't really optimize against that. Well, actually, we did, because let me show you here in the model editor for ADMET risk. You can actually see this is kind of a composite function that is made of several objectives. And if we look at the risk rules, which I will put large here, you will see that ADMET risk is made of 24 parameters, 24 different objectives, one of which, actually line 8, is solubility. So we did include solubility as part of our target functions, if you will, or target objectives as part of the optimization. So again, so we've been able to achieve multiple objectives here in optimizing this set of compounds. So I think that's where I'm going to stop and hand the controls over to back to Michael.
Okay. Thank you, Eric. We appreciate that presentation. It was very good. We have some questions already, but we're going to hold off until after David's presentation to discuss those. So just go ahead and type in your questions now if you can, and we'll answer them after David's presentation. Before we get to David's, I want to launch one more poll. So I'll go ahead and launch it. And this one is based on what you've seen so far. Do you believe ADMET Predictor could be used to support your medicinal chemistry efforts in your organization? So we're getting in some votes now, and we appreciate that. We've got about 10 more seconds, and then we'll turn it over for David's presentation. All right, thank you very much. And now I'm going to make David the presenter. Take it away, David. We see your screen.
Okay, thank you, Michael. All right, so now we're going to switch gears and focus on a somewhat later stage project for this demo, compound design. So we're going to be designing inhibitors of an enzyme called beta secretase 1, or BASE1. And these inhibitors should have good predicted activity and ADMET properties. And we're going to try to have them be structurally similar to some lead compounds that were taken from a relatively recent MedCam article, which I'll show. So the actual steps of the demo will be to build a BASE1 activity model. And we're going to supplement the MedCam data with data downloaded from the Chembl database. We'll then generate some structural analogs of these MedCam compounds, and then evaluate the analogs using both predicted ADMET properties and predicted BASE1 activity using the model that we build. Okay, so I have ADMET Predictor open, and the first thing I'll do is open up the compounds from the MedCam article. So we extracted those compounds. I have them here in an SDF file. There are only 15 compounds here. You can see if I scroll through them that they are very structurally similar to one another. There's a column here with a link to the original article as a PDF file. If I click on that, we can open up the file, and they've drawn the scaffold for these compounds, and they've specifically indicated that there's an amidine substructure contained in this ring system, and they identified that amidine group as being important for inhibition of the enzyme. So when we design analogs, we will ensure that all of the analogs that are designed have this same functional group. Okay, I'll close that. The compounds all, of course, have experimental BASE1 PIC50 values shown in this column, and I can put those into the histogram as well. So the range is about 5.5 to 8.2. Okay, so I mentioned the next step would be to supplement this data with some Chembl data so that we can go ahead and build an activity model. So I've already extracted those compounds from the Chembl database, so I can import those. So I have another SD file for that. Okay, so those get brought into the program. You see the histogram of activity values has been updated. The range is now 3.7 to 9.7. I should say that ahead of time, I went ahead and eliminated any of the Chembl compounds that did not have that ring amidine functionality. As I mentioned, we've decided that the analogs we generate will have that group. So it makes sense that when we build a QSAR model, that we can restrict the model to being built only with compounds that have that group in it. I can just quickly do a substructure search to confirm the presence of that group. I could launch Designer here. Michael alluded to that. I could draw the substructure, but I have it encoded in a mol file here. Amidine, with these annotations indicate the ring requirement. If I search with that, you'll see that every compound in the set of roughly 400 compounds has that group. Okay, so with this data, not a huge data set, but about 400 compounds, we can build a model of base 1 PIC50. Let me just show a quick slide to illustrate the workflow for model building inside ADMET Predictor. So starting with structures and experimental data, the first step is to calculate descriptors to use for model building. And ADMET Predictor has on the order of 350 proprietary molecular and atomic descriptors, and I'll calculate those. We've described those in past webinars, and the user manual has a lot of information about those. At that point, we launch the separate ADMET Modeler tool that Michael alluded to. The first step there is to divide this data into a training set, which is actually used for the model building, and a test set, which is used only for evaluating the models. It's an independent test set. We build many models, some relatively simple, some more complex. ADMET Modeler automatically chooses a best model that should have reasonably low complexity and importantly, good performance on both the training set and the test set. So I'm going to illustrate this entire process here. So the first step is to calculate the descriptors. Eric went through this workflow earlier. In this case, I don't need the ADMET properties, the phys, chem, metabolism, etc., just the descriptors. So I'll launch that. Fast on this set, of course, and Eric alluded to the planned speed ups to this. Okay, so now we have our molecular and atomic descriptors for the 400 compounds. So we can launch the ADMET Modeler tool. We just need to specify a dependent variable. So I'll choose base 1 PIC50. Then the modeler interface opens. I should just point out that Modeler is obviously the tool that customers use to build predictive models using their own data, but it is also the tool that we use internally for all of our commercial ADMET models. The dependent variable is shown here, the base 1 PIC50. All of the descriptors that we calculated are selected here automatically. There are a number of other parameters that we won't be able to talk about today because of time. There is a useful option here to import parameters. Typically, these are saved from a previous run. And I'm going to quickly do that just to ensure consistency with my previous results. And then I'll just talk about a few parameters here. If I advance to the next page, you'll see the default model type is neural network. Of course, in this case, we're building a regression model. This is real value data. If you have, yes, no, or a toxic, non-toxic type of data, you can also build a classification model. The test set will be chosen automatically by Modeler. The default size is 10%, and here the method is stratified sampling, which means these compounds will be sorted by the dependent variable, the PIC50, and then compounds will be selected randomly within the different activity bins. So that way the activity distribution of the independent test set will match that of the training set. There are other parameters here, and we've talked about some of these in previous webinars, and of course our user manual has complete information. But here I'll just start the model building process. This grid looks like what we saw on the slide. So here the rows are number of neurons in the hidden layer of the neural network. The columns are number of descriptors. So we're building some relatively simple neural network models, and as you go down, they are more complex with more neurons and more descriptors. Modeler will highlight what it thinks is the best model, again, reasonably low complexity and good performance on both training and test. If I click this box, you see the scatter plot. So this is predicted versus observed base 1 PIC50. The blue points are the training compounds and the red are the test compounds. So here the performance is roughly the same for the two. There's a lot of other statistics that are shown here that you can look at. In a real project, of course, you would spend typically a lot of time building models, looking at outliers and going back to the original, references where the data came from, if this is literature data, and perhaps, excuse me, perhaps experimenting with other modeling parameters. For this short demo, we'll accept this model. It's not bad, given that it's Kemble data in particular. If you're satisfied, you can right-click on a model and export it, which then makes it available to ADMET Predictor itself. So I'll provide a name, call it base 1 model, and then close modeler. ADMET Predictor is now aware that there is a new model, and we can use this model now to make predictions on new compounds. Okay, but the next step is going to be to generate some analogs, some structural analogs of the original MedCam compounds. Let me just show one more slide to just talk a little bit about the methodology here. So one of the methods in Predictor for generating analogs uses what we call transform rules. This feature is inspired loosely on a tool that was described in a 2006 article, and this was from Abbott Laboratories, and they described a product called Drug Guru, where you would take a lead compound and apply to it a database of transform rules. And in that case, the rules were actually real reactions that were used in-house within Abbott. And this was a way to, generate potential analogs and give chemists an idea of where they might go synthetically from their starting compound. So we've generalized this so that our set of rules includes things like bioisosteric replacements, some actual reactions, and then some what you might call generic mutations, things like change this atom type to a different atom type. And that allows you to have a broader chemistry space that you can explore. Of course, users can modify these rules. You can modify our rules, add your own rules as you like. Okay, so I'm going to take one of the MedCam compounds. They're all, of course, very similar, and use this tool. So there are, again, a number of parameters here, and I'll focus on just two of them. One is essentially the number of generations. So one generation would mean take the lead compound and apply the database of transform rules just one time. Two generations would mean take those analogs from the first generation and apply the rules a second time on those analogs. So we'll set this number to 3. Obviously, the larger this number is, the more analogs you can generate. The second thing is we call filters. So you can supply some kind of arbitrary queries to restrict the types of analogs that you produce. So I've encoded some of these rules in a file called analog drug-like filters. So this file, on the one hand, contains some fragments that you typically would not want to see in a drug molecule. It also has a restriction that each analog must contain this ring amidine motif that we were talking about earlier. Then I'll also check the box that just says we don't want to produce an analog that we already have in the spreadsheet. So it'll ensure that everything is novel with respect to what we already have here inside ADMET Predictor. Okay, so if I launch that, it'll run. And again, what we're doing is generating some structural analogs of a lead. And we can then profile those analogs using our ADMET predictions, as well as our base one activity model. I wanted to point out that one thing we're working on for the next version of ADMET Predictor is a way to automate this design process. It's going to use an evolutionary algorithm where you generate a smaller number of analogs, calculate their properties automatically, weed out the less optimal compounds, and then repeat that process iteratively. And you can go through a lot of generations relatively quickly that way and directly optimize properties. Users will be allowed to select the properties that they want to optimize. And they can include our ADMET properties, activity models that you build, as well as our HTPK properties, like fraction absorbed and fraction bioavailable. So that's going to be a forthcoming feature. Okay, so that process completed. We did cap the total number of analogs at 10,000. I don't know how many this would have produced without that cap, but you can imagine that this feature can generate a vast number of structural analogs. The very first compound is our starting compound, the lead compound. And there's a column here that contains the set of rules that were used to produce the analog. So there's names of the rules taken directly from that transform database. So you can look at these and get an idea of how the particular analog was produced. So our next step would be to predict properties of these analogs, both ADMET and our base one activity. And I have those saved because there are 10,000 compounds here. That would take about 5 minutes on this machine. So let me just open up a saved file. So the same 10,000 compounds, but with all the ADMET and base one activity already calculated. The predicted base one activity is shown here in the histogram. So predicted activity ranges from about 4.1 to 9. In the scatter plot, that same activity is shown on the X, and the ADMET risk property that Eric was alluding to is shown on the Y. So again, that's kind of our overall ADMET liability score, with lower values being better. Okay, so our next step would typically be to kind of weed out the less desirable of these structural analogs. One way to do that is with a query. I can actually use the same substructure search feature. With that feature, you can also specify property filters. And I have some in a file here. And you can just type these in the interface. So it says, for instance, our base one model, the predicted PIC50, if it's below 7, then we're going to consider that an undesirable compound. Similarly, ADMET risk greater than 6. Because base one is a brain target, if the predicted brain penetration is low, we'll eliminate the compound as well. So if I do that, that query, I can just eliminate those compounds from the spreadsheet. Okay, so now our predicted range of activities is much narrower, 7 to 8.5. Same with ADMET risk. Now it only goes to 6. If I select our original lead molecule here, this is the MedCam compound, I can show its position in this scatter plot. So you can see its ADMET risk is actually not so bad. But there are compounds, for instance, these out here, you might say, have both higher predicted base one activity as well as lower overall ADMET risk. I can just show where these compounds are in the spreadsheet. Here's the first of them. This compound, you can see, has it's a kind of a cyclic derivative of the original compound. There's some other changes as well. As to whether this compound would actually be active against base one, we don't know that. We only know that it was predicted to be active in our base one model. Of course, that was, kind of a quick and dirty model built using a pretty small data set. But hopefully you can appreciate the workflow that we went through here, where we pretty easily took data, generated an activity model, generated analogs of some lead compounds, and then profiled those analogs using the predicted properties. And again, I think the important thing to emphasize is that this is really an idea generation tool. So you can look at the kind of high priority analogs and get an idea of where you might go synthetically. Okay, there's a few minutes left. I wanted to show one more feature just briefly. This has to do with similarity searching, very common cheminformatics feature. In this particular workflow, one use case might be, taking a compound like this and checking whether it might be available in a commercial database, like a vendor database. So ADMET Predictor has a feature to search an external library of compounds. So you would generate that library by taking an SD file or a SMILES file and pre-processing it into a special format that would allow it subsequently to be searched very, very quickly. So if I select this compound, I can go to library. You can also do an exact substructure search. Here we're doing kind of a loose similarity search. You can see I have the entire Chembl database here, which is now in searchable format. There's a company called Enamine that has, this is a chemical vendor company, and they have a database of about 800 million compounds that represent things they believe they could quickly synthesize and deliver. For purposes of this demo, I took a 5 million compound subset of that 800 million database and created this searchable library of it. And so we can very quickly search those 5 million compounds. This is using several processors, but that's increasingly common these days to have a multi-core machine. So to search that many compounds, it's very fast, both for similarity searches and for substructure searches. Of course, you could do this with a patent database, your own corporate collection, etc. This is our starting compound. These were the closest matching compounds found in that Enamine database. What I wanted to do was say that there's a nice way to view these so that you can really see the structural differences between the query structure and the database structures. And that is to use this pairs tab, which is meant for looking at things like activity cliffs in a data set. In this case, you can see here on the left is our query structure from the MedCam paper, and here are the closest matching analogs from the Enamine database. And you can see that what's nice about this similarity feature is that it'll actually, knows sort of what the common scaffold is between these two compounds, so it can actually align them and highlight the exact atom and bond differences between them. So it's not just a kind of a vague Tanimoto similarity. It's actually looking at the the exact atom and bond differences between the compounds. Of course, here we don't see too many very close analogs in the Enamine database. Kind of the overall topology we can find, nothing with the amidine. But in any event, hopefully you could see that this would be a useful tool for doing very fast searches against a pre-processed database, either your corporate collection, patent database, vendor catalog, Chembl, etc. Okay, my watch shows 5 minutes to 9. I think we wanted to stop at this point to see if there would be any questions. So yeah. I'm going to hand the controls back to Michael.
Okay, you can maintain the presenter because we've got a few questions. Before we answer the questions, I just want to launch one more poll. So let me just launch that quickly. Based on what you saw today, how likely are you to request an evaluation of ADMET Predictor? And so we'll just give you a few seconds of that, and then we'll start addressing the questions. So we have a few questions. I just want to mention that one of the authors of Drug Guru, Kent Stewart, is actually attending this, and he really liked what he saw in your presentation, David. So very nice compliment from Kent.
Oh, great.
And so let me go ahead and close that poll. Okay, Dave, the first question kind of refers to R tables in Eric's presentation. And the question is, can you export the R table?
Yeah, there are a few ways to export it. I think the most common one is as an Excel file, where you get the, all the structure pictures and all of that. You can of course export the compounds themselves as an SD file, SMILES file. I think there is also a way to export the R groups themselves. There are various design tools in ADMET Predictor where you can make use of that list of R groups. I think the attachment point on the R groups is designated somehow in the exported file, and you can actually make use of that.
Excellent. So you would, you'd be in the R tables tab, and then you would say File Export, and then export to different format.
Yeah, I believe so.
Okay, excellent. The next question is, can you show how to get a pH solubility profile of a compound? So I think if you select a compound in one of those files, and yeah, just click on the, have you already calculated ADMET? So go to the, oh, you didn't calculate the PhysChem properties.
Yeah, my screen is, for whatever reason, is giving me a hard time right now.
Okay.
But there is a way to do it. I don't know if you can see my screen now.
Yeah, we see it. We see it fine. Yeah.
Okay, yeah. So I mean, there are options for solubility versus pH, log D versus pH, which will give you the curve. I think Eric just showed that.
Yeah. You just double click on that cell for that compound, and
there's also a very
it opens up the window. Opens up a new window.
Yeah, there's also a very nice pKa microstate display where you can kind of look at the population of various ionization states as a function of pH. We didn't have time to show that.
Yeah,
excellent. So another question is, can the software predict panes or use panes filters?
Yeah, I mean, there's a very flexible query engine inside. So I don't know that we distribute a file that has the panes definitions in it, but those are easily obtained from the internet, and the software has lots of tools where you can add a score to your compounds that indicate how many panes violations there are. You can use those to weed out compounds you don't want during a design phase. So yeah, anything related to to queries, there's a lot of support for that.
Excellent. And the drug-like filter you applied, if someone wanted that, they could contact you via email and you'd send that to them. Yep. Excellent. Okay, so another question is that they're using version 9.5, but some of the functions you've talked about today aren't available. And I think it's just simply getting a license for that module. So a lot of what we've displayed, in the R tables and things like that, clustering compounds, that's in the MedCam Studio module, and we'd be happy to provide a 3-week evaluation of that module if you're interested. So just contact either me or Eric at simulations.plus. You can also use the info@simulations-plus.com to submit that. All right, so the final question we have is, can we get the pharmacological activity of a photo switch compound which is light sensitive? How accurate is ADME? So that's not one I'm familiar with, pharmacological activity of a photo switch compounds.
No, I admit I'm not either.
I don't know. I really don't know.
Bob is, I think, unmuted. Bob, if you care to join the chief science.
Several different kinds of photo switch compounds. Usually it's going to be an EZ flip and a double bond. And those we are generally not going to differentiate. On the other hand, what we have found, and also what GSK found, is it for off-target things? Typically, the effect of things like that are pretty small. So for toxicities, it would be a little surprising if one was much more toxic than the other, if it's an off-target toxicity. Is that adequate for the whoever asked?
Yeah.
I'm not sure. Yeah, we'll see. They can contact us. I did. There is one. Oh, it says okay. Yeah. Thank you, Bob. There was one more question, Dave. Can the similarity search give you a similarity score, such as a Tanimoto?
Yeah, the similarity search does give a similarity score, and it is in the form of a Tanimoto. It's just that it uses, kind of, as I was saying, the exact number of mismatch atoms and bonds between the two compounds to, those are kind of combined together into what looks like a Tanimoto score between 0 and 1. So you can rank the compounds by similarity, but it's a little different than what people often, think of as a fingerprint-based Tanimoto score, where you don't really have a good sense of, what how big the common core is between the two compounds. I think it's a better method, more chemically intuitive.
Excellent. Okay. Well, that was some good questions from the audience. So I think we're just a few minutes over, so I just want to conclude this unless Eric or Dave, you have anything else to say?
Nope.
No?
Okay.
Michael?
Okay. Well,
that concludes our today's webinar. If you missed any part of today's presentation, the playback will be available at our website, www.simulations-plus.com. So thank you all for joining us and have a wonderful day.
All right.
Bye-bye.
Thank you, Michael.
Bye-bye.
Bye-bye.